So, I went back and looked through my files, and I was down there 2 years ago giving a talk. And let me tell you, it's a little challenging to give you a new talk in 2 years. I should be doing better than that, but things don't change that fast. Not to mention that the last year, we didn't really collect any data, right? I don't know about you, but we didn't do a single experiment for like 14 months. So I had to really think about what I wanted to talk about today. And I decided I was going to talk about what's happening, what's happening to cognition and memory, and how Alzheimer's disease comes about more than like clinical things. Now, of course, I don't know how Alzheimer's disease comes about, so that's probably the biggest caveat I have to give you, but I'm sure you will forgive me for that. So I'm going to show a few slides that I showed 2 years ago. I can't help it, but most of what I'm showing is either I haven't shown you or is new, and it's either just been published or coming out. So those are the disclosures. So I don't really have to spend much time on this. You all know we can measure beta amyloid. Most recently in plasma, the first way we measured it, of course, was in, not us in my lab, but it was measured in CSF. And it's kind of interesting to me that CSF, I think, sort of never really caught on until PET came along, when we realized that all the false positives from CSF weren't false positives at all. They were true positives, that just a lot of people have amyloid. So that's the A in ATN. The tau, same story, CSF first, then PET, and more recently plasma, and that's the T. And then neurodegeneration, which of course we can measure with MRI, FDG, and in a number of different biomarkers in CSF and plasma. So I'm not going to talk very much about this ATN. I just wanted to sort of put it out there. And most of the data I'm going to show you is using these biomarkers as continuous measures, not dichotomous measures so much to classify people. So I'm going to talk about data from two cohorts. One is the cohort we're following in Berkeley, a Berkeley Aging Cohort Study, or BACS. This is a convenience sample. It's not representative of aging, unfortunately. It tends to be fairly healthy, fairly white, fairly affluent. But we have a lot of data. We have 260 baseline amyloid scans and 160 baseline tau PET scans, and we've got a fair amount of longitudinal data. So I'm going to show you a little data from BACS, and then also some data from ADNI, which I think, again, you all know about. ADNI isn't particularly representative either, and we're making efforts to try to change that in the next cycle of ADNI and ADNI 4. But there's a tremendous amount of data in ADNI as well. So I'm going to flip back and forth between these two cohorts and not spend a lot of time talking about them. So, we've known that we can measure amyloid pathology for years. We've been able to measure it in CSF and PET. And probably one of the most interesting phenomena that kind of got people thinking very early is that the relationship between amyloid and cognition is very weak. And if you look at that, either pathologically or just the fact that 30% of cognitively normal older people have amyloid in their brain, measured with PET, and they frequently have quite a bit of amyloid in their brain, and yet they're completely cognitively normal. It's pretty clear that this is not a strong relationship. But I think over the years, we've kind of come up with this model of Alzheimer's disease, that it's really not amyloid itself, but it's some interaction between amyloid and tau that causes the tau to become pathological. It causes the tau to spread throughout the brain, which in turn is related to inducing these neurodegenerative changes, which then have the strongest effect on cognition. So this cascade of events, which has been promoted as the amyloid hypothesis in a nutshell, has been one of the guiding models of Alzheimer's disease for 30 years or so. And it's still controversial. I think everyone knows that. And usually in my talks, I spend about half the talk going over the evidence for this model from the imaging perspective. And in my view, there's quite a lot of it. But I want to just tell you about one paper that we published this year that, to me, starts to make pretty good evidence in support of the model, because it's longitudinal data. And this was a paper that came out at the end of last year in Biological Psychiatry from the ADNI data. And in this analysis, we looked at the bilateral relationships between all of these biomarkers. Actually, what I'm going to show you is not cognition, but let me take you through this. So in each of these graphs I'm going to show you, the independent variable is up here. The dependent variable is here, and this is the rate of change in the dependent variable over time. So what we're looking at here is the effect of fluortaucipir, this is tau, on amyloid. And you can see that there's a level effect, but there's no interaction. In other words, there's no effect, there's no difference in how quickly high, medium, or low tau patients, or these are cognitively normal and impaired individuals. There's no difference in how these three groups of individuals deposit amyloid. So in other words, from this graph, there's no effect of baseline tau on the rate of change of amyloid. And there's no effect of baseline hippocampal volume on the rate of change in amyloid. So, to me, this is an important finding. We have only about an average of about 2.5 years of follow-up in these individuals, but the pretty decent samples, and there's no evidence that tau drives amyloid from this particular analysis. When we look at amyloid and tau, though, we begin to see the interaction. So the high amyloid individuals are depositing tau faster. So there is a significant effect of amyloid on tau. Again, no effect of hippocampal volume on tau. And then finally, we're looking at hippocampal volume. There's a small effect of amyloid on hippocampal volume and a much larger effect of tau on hippocampal volume. So amyloid drives tau. Amyloid, to some extent, drives hippocampal volume, and tau drives hippocampal volume. But there's no evidence that this process proceeds in the reverse direction. So to me, this is pretty strong evidence, again, favoring the amyloid hypothesis. So that's really all I'm going to say. And now I'm going to go into these three aspects of my talk: what mechanisms drive amyloid, what mechanisms drive tau, and what are the consequences of tau deposition. And obviously we don't know the answers to this, so what I'm going to be talking about is pretty speculative. So there's been a fair amount of human data, and interestingly, this whole slide is a bunch of different studies, and they're all from Washington University. So Randy Buckner has proposed that this is something about the default mode network, but I think pretty soon after he came out with that idea, he focused on this idea of cortical hubs. These are areas of the brain that are highly connected. And he pointed out in both of these papers that these highly connected or highly sort of metabolic areas are depositing amyloid. And what these have in common is this idea that there's something about neural activity or metabolism that's related to amyloid. Mark Rakel's group published a couple of papers. They've published a series of papers now on this idea of aerobic glycolysis. What they found is these are brain regions in red, where the brain is metabolizing more glucose than it should, given that there's oxygen present. So it's very inefficiently metabolizing, and that's because the glucose is being metabolized down the glycolytic pathway rather than the oxidative pathway. And so it's just burning glucose when it really doesn't have to. This is something that is called the Warburg effect. It's been found in cancer tissue, and it's not clear why this happens in the brain. It may have to do with the speed of glycolysis compared to aerobic metabolism. But at the end of the day, these are brain regions where glucose is sort of overutilized that are also depositing amyloid. And then there's been a tremendous amount of data. This is one of the earlier papers from John Cerrito, again at WashU, that showed basically in a very beautiful, elegant experiment that synaptic activity releases beta amyloid, that at the synapse there's beta amyloid release. And so there's been a lot of data in animal models suggesting that neural activity itself releases beta amyloid. So all of this together really points to the question of whether it's something about metabolism or neural activity that drives amyloid. So one of the first studies we did looking at this was done by Hwami Oh, who was a postdoc in the lab. She's now at Brown. And what she did is she decided to look at the patterns of amyloid across the lifespan, of glucose metabolism across the lifspan. Of course, you can't follow people for 70 years. So she took a group of young subjects, a group of PIB negative old, PIB positive old, and Alzheimer's disease, and looked at their glucose, their FDG PET scans at baseline. And so she looked at the brain regions that were highest in young people. Those are these brain regions, not terribly important where they are, although they are in areas like posterior cingulate, precuneus, medial prefrontal cortex, maybe beginning to sound a little familiar to you. These are the brain regions that are highest in glucose metabolism in young people. And then she looked at the brain regions that are lowest in glucose metabolism in young people. And she looked at what they were looking like in these older individuals. Again, completely different samples. So these are the comparisons of FDG in the young PIB negative, PIB positive, and Alzheimer's cases. And of course, this is circular reasoning. The high metabolism in younger adults was higher than the low metabolism in young adults. That's just a circular reasoning. But this held through the lifespan. So even in the PIB negative older subjects, the brain regions that were high in young people were higher than the brain regions that were low in young people. So more metabolism. So this suggests that this metabolic difference persists through the lifespan. And it was even the same in the PIB positive old subjects. So this is just to show us that this metabolic difference persists throughout the lifespan. It disappears in Alzheimer's disease, not surprising, because metabolism goes down in the high and the low regions. So the real question is, where does amyloid deposit? And the hypothesis was that amyloid would deposit in the high metabolism brain regions. So this is now PIB. And what you can see is that in the PIB positive subjects, the brain regions that showed high metabolism in youth deposit more amyloid than the brain regions that show low metabolism in youth. So this is how these brain regions were selected. They were selected based on metabolic rates in the young subjects. And there is a substantial difference in the amount of amyloid in the high metabolizing regions compared to the low metabolizing regions. So this was one of the first papers that we did that suggested that glucose metabolism is related to amyloid deposition. Stephanie Leal, who also was a postdoc in the lab, now at Rice, went back and looked at one of actually it was the first fMRI study we ever did in the lab. And actually Beth Mormino was the one who did the study, and she tracked down all the participants and got their subsequent PIB scans. And so that's what you're seeing here. 0 is the baseline when they had their functional MRI scan. So this was a memory encoding task. I'm not really going to talk about it, but she simply looked at the amount of activity in the cortex during memory encoding at baseline, and then looked at the change in amyloid over time. And you can see that lots of these folks are actually depositing amyloid. These red lines, all the red lines are positive slopes. The blue lines are people who are not depositing amyloid. So these folks are depositing amyloid over time. And she simply asked the question, what's the relationship between functional MRI activity at baseline and the rate of amyloid accumulation over time? And if you look at the hippocampal activation, it was positively correlated with the slope of amyloid, but actually non-hippocampal activation wasn't. And this gets into a whole nother story of whether activity in the hippocampus and the cortex are different. And I'm not sure I know the answer to that, but at least here they seem to be. But the bottom line is neural activity in the hippocampus was associated with more amyloid deposition over the next 3.5 years. So say something else about amyloid. We published this paper just a couple of months ago looking at ADNI. And this graph shows the relationship between amyloid at the baseline PET scan and the rate of change of amyloid over time. Now, there are a lot of PET scans in this study. There were about 1,600 PET scans in about 700 people. So everyone had 2 to 3 PET scans, basically. And what you see is that at low levels of starting amyloid, the rates of amyloid accumulation are very slow, but then they really speed up, and then they sort of hit the peak, and then they start to slow down. So there's a quadratic function associated with rates of change of amyloid, and this has been why this is a replication of other reports that have come out of the Australian study, the Mayo Clinic study, I think a couple of other studies, but those are the 2 that stick in my mind. Actually, we see it in our data in Berkeley. I'll show you in a minute. So this inverted U quadratic function has been pretty well replicated, that amyloid rates seem to slow. And what's interesting about this is if you simply do a time integral of this, you can come up with a graph that has time and amyloid on the X and Y axes. And when you do that, you see that the time from the typical amyloid negative normal is about 7 years. And then to the peak is another 10 years. And then subsequently, another 10 years to the typical level of an MCI, and another 8 years to a typical level of an Alzheimer patient. So this time period is really the time period for primary prevention, but it's very early. It's very early because amyloid starts to slow even before people develop symptoms, at least in this data. Again, another 10 years before people become impaired. And this is really the key point, that this peak of amyloid is before the level at which people hit mild cognitive impairment. So this, to us suggested that if you're going to lower amyloid, lowering it in people with Alzheimer's disease and probably even MCI is pretty late, because at this point amyloid is slowing. And I think one of the things that I think is very interesting is the possibility that amyloid has induced some change in tau that at some point becomes independent of amyloid, because the amyloid is slowing, but things are getting worse. And so what this suggests to me is that something's happening with tau that no longer depends on amyloid. But that's just obviously conjecture.
Bill, so quick question on that.
Yeah,
those levels, those people are not yet called positive, or are they called?
They're positive. Yeah, they are. They're positive at about 7 years, and then it peaks after they're positive. So it peaks after the positivity threshold.
Okay,
sure.
But it'd be a tough read, probably. If you were reading it clinically, it'd be just
25
at the edge.
25 centiloids is pretty low. In our data, this is positive, but most visual reads are between 25 and 35. So,
okay, thanks. Interesting.
Yeah. So we looked at this, I'm showing you this for a few reasons. One is we're just talking about amyloid accumulation, but this led us to think about this approach to try to get a sense of how amyloid spreads through the brain. So what we did, and we went back to our Berkeley data, where we have a lot of nice regional data, and we fit all these different brain regions to this inverted, to this quadratic function. So here I'm just giving you a smattering. This is this is a totally different data set now. This is the data from Berkeley. I think you can see a really nice inverted U in these data, by the way, and I think that's because we measured distribution volume ratios. We collect 90 minutes of PET data, and I think those are really nice for longitudinal studies. But I think, anyway, whether you agree or not that these are, sort of tighter data than the ADNI data, we could find this inverted U in multiple brain regions. And so Molly Lapointe, who is a graduate student in my lab, looked at this in multiple brain regions, and that's what you see here. And these are all the brain regions. This is the global PIV value when each region hits its peak. And you see they're pretty close. These are the mean slopes. They're pretty close. These are the centiloid values at the peak. They're pretty close. But this is really what I want you to see. What time does this region hit its peak? And they all hit their peaks within about 4 years of each other. So there's no real evidence that amyloid it doesn't seem very likely that amyloid starts in one brain region and spreads to another. They all peak within a couple of years of each other. So I just tied this to some data from Michel Grote, who is in Germany, who did a study where he looked at the gene expression profile of different brain regions in the Allen Brain Atlas. And what he basically found is that in the brain regions where amyloid is deposited, there were two clusters of genes, one of which was associated with immunity and protein synthesis, and the other with mitochondrial respiration. So these are the genes. This is a lot of data. It's not my field. You can look this up in the original paper. But one of his conclusions, which really appealed to me, was the idea that that the brain regions where amyloid is deposited are expressing genes involved in metabolism, whether that's protein synthesis or mitochondrial respiration. And again, this ties back to the idea that there's something about this tissue that makes it vulnerable. So it's not the idea that amyloid is spreading from one region to another. It's more the idea that amyloid, the way I think of it, is amyloid is blossoming throughout the whole brain based on tissue vulnerability, something about the tissue where the amyloid is deposited. One last experiment, again, that talks a little bit about this vulnerability is an experiment we did with Matt Walker, who is a sleep scientist at Berkeley, and Bryce Mander, who was a postdoc that Matt and I both supervised. Bryce is now at Irvine down in your neighborhood. And Bryce and Matt made the observation that the amyloid pathology is deposited in the same parts of the brain that generate slow wave sleep. So they came up with a clever experiment where everyone had an amyloid PET scan. This is our Berkeley cohort. In the evening, they studied word pairs that they had to remember. They slept in the sleep lab where they had a polysomnogram. And then after sleep, they were tested on the word pairs while they were in the magnet. And so the first thing that they found is that the more amyloid these individuals had, the less slow wave activity they had. And it's particular slow wave activity, less than 1 hertz. So this shows you that as amyloid goes up, slow wave sleep goes down. The next thing that they found is that the more slow wave activity, that the less slow wave activity they had, the more the hippocampus was engaged during retrieval. So this we think of as a bad thing, because as your memory is consolidated, the hippocampus should become less and less important to retrieve memories. And so when these subjects were in the fMRI scanner, retrieving the word pairs, the less slow wave activity they had, the more they needed their hippocampus to do the task. And then the more the hippocampus was activated, the worse they did on memory. So this again suggests that another story in terms of amyloid deposition is sleep. And we really think that this may be a bidirectional effect, that that beta amyloid has an effect on sleep and metabolism, and the sleep dysfunction leads to amyloid deposition and to increased neural activity. It changes the neural activity. So there's something about sleep dysfunction that's driving amyloid, and the amyloid could be driving this change in neural activity. And so it's a vicious cycle. And the same may be true for lifelong metabolic changes. So there's really a number of things that are driving amyloid, but they all seem to be related to some kind of neural activity. So where does this leave tau in this story? So the first thing that strikes you when you look at tau in relationship to amyloid is how differently they're distributed. So this is a heat map showing you the correlations between multiple regions of where amyloid and tau is deposited. So let's start with this. So this is PIB, this is amyloid. So let's look at amyloid in the left cingulate in terms of its correlation with tau in all of these brain regions. Okay, so we start here in the left cingulate, not much. The left frontal, yes, there's a bit of correlation. Occipital parietal, not much. Subcortical, not much, but temporal. So amyloid in the left cingulate is highly correlated with tau in the left temporal lobe, and same thing for the right temporal lobe. And I don't think I have to go through every one of these rows to convince you that no matter where the amyloid is in the brain, it's correlated with tau in the left and right temporal lobe. And so amyloid anywhere in the brain is correlated with tau in the left and right temporal lobe. And if you just map this out, this is what you see. So these hot areas, these are just partial correlations for every pair of ROIs. That everywhere you look, the amyloid is correlated with tau in these reddish orange brain regions, which are largely lateral, inferior temporal lobe. And I think inferior, lateral, and anterior temporal lobe, much more strongly than anywhere else in the brain. There's some correlations, in the parietal and frontal lobe, not much in the sensory motor cortex, but really inferolateral temporal lobe. So how does that happen? So obviously, again, we don't really know how this happens, but one of the major theories of tau spread, of course, is that it spreads through neural connections, and there's been a tremendous amount of animal work to show that tau seems to be able to spread across a synapse, that it's released and taken up by the postsynaptic neuron. And there's a lot of interesting data in humans looking at functional connectivities to suggest that brain regions that are more connected to one another are more likely to co-vary in the amount of tau in them. So we were very interested in this idea. This is just a sidetrack. This is some really cool study by Renaud Lajoie, who showed that one of the things that seems to drive tau into the temporal lobe is the ApoE4 allele. And this has been widely replicated, actually. It's probably one of the most replicated studies with tau, is that the E4 carriers have more tau in the medial temporal lobe. And the case, these are Alzheimer's patients. And the Alzheimer patients who don't have E4 are more likely to have tau in the neocortex. But that tells you maybe a little bit how it gets in the medial temporal lobe. The question is, how does it get out of the medial temporal lobe? So to do that, a grad student in my lab, Jenna Adams, who's now a postdoc actually at Irvine, looked at functional connectivity. So functional connectivity, I think you all know, is simply looking at the bold signal in different brain regions and looking at its synchrony. And areas of the brain that are highly synchronous, that are highly correlated, are inferred to be connected to one another. So I'm going to take a little digression here and talk about the memory system. And there's been a lot of interest in the idea that the memory system can be sort of parcellated in a number of different ways. And there's still a lot of debate about this. But I think Charan Ranganath at UC Davis has been a major proponent of the idea that you can think of the human memory system as subdivided into two networks. And one of these networks is an anterior temporal network that is specialized for memory for objects. And so that network has a sort of a hub in the perirhinal cortex, and it's connected up to the hippocampus, but it's also connected up to the lateral orbitofrontal cortex, the ventral temporopolar cortex. And this is a network that involves a lot of these anterior temporal brain regions. And then there's the posterior medial network that is more specialized for spatial information. And that hub is sort of the parahippocampal cortex. It's also connected to the hippocampus. And in fact, this is the brain region where the object and spatial information come together. So this posterior medial network is functionally distinct and spatially distinct from the object anterior temporal network. So why am I spending your time on this? Well, it's because where tau deposits in the medial temporal lobe is very interesting. And these arrows point to what Brock has called the transentorhinal cortex. And it's actually transentorhinal cortex because of the architecture of the layered cortex. But it's interesting because it happens right here, which is the borderline between the perirhinal cortex. So this is perirhinal cortex here, and the lateral entorhinal cortex here. And this is the medial entorhinal cortex. So that's the anterolateral entorhinal cortex, and that's the posterior medial entorhinal cortex. And we know these areas are quite different, have quite different specializations, right? And the posterior medial cortex and the anterolateral cortex are wired up quite differently. And in fact, the anterolateral entorhinal cortex is connected to the perirhinal cortex, so it's part of this object network. And the posterior medial entorhinal cortex is wired up to the parahippocampal cortex, so it's part of the spatial network. And this is where the grid cells are, for example. So these are two very different networks. Now, just parenthetically, I want to say that the posterior medial network looks like this, and this is where the beta amyloid is deposited. And the anteromedial network looks like this, and this is where the tau is deposited. And it's remarkable to us, and we wrote about this in a paper in Brain that Anna Maas was the first author of. And I think there's just tremendous evidence that the spatial distribution of these two pathologies follow different networks. But I'm not really going to specifically get into that, because what I really want to talk about is this anterolateral entorhinal cortex. And the experiment that Jenna Adams did was to say, well, we think that the areas of the brain that are connected to anterolateral cortex should be the parts of the brain where most of the tau is. And that's because this is where the tau starts, in the anterolateral entorhinal cortex. So what she did is she took data from young adults to create these networks, and she tried to pull out the anterolateral connected network and the posterior medial connected network. And these are the data from her paper that shows the anterolateral network. And again, if you remember where I showed you where the tau is, it really looks like the parts of the brain that tend to deposit tau. And there's the posterior medial network. Again, looks a little bit more like the brain regions that tend to deposit amyloid. And this is the overlap, and there isn't much. The purple areas are the areas that overlap. So I was pretty shocked that she was able to do this, to be honest with you. But it was probably because she used an atlas that was developed using 7 Tesla data. And she was just able to do this very fine spatial localization. So she pulled out these 2 networks and she simply asked the question, since tau deposits in the anterolateral entorhinal cortex, these brain regions that are connected to the anterolateral entorhinal cortex should have more tau than the brain regions connected to the posteromedial entorhinal cortex. And, you know, I mean, I wouldn't be spending all this time if she didn't find that. So this is the actual data read. So now we're looking at cortex. Now we're looking in the connected brain regions. So these are brain regions that are connected to the anterolateral, functional connectivity to the anterolateral entorhinal cortex, posteromedial, and then outside. And you can see that across all the older adults, this was true. Even in the amyloid negatives, it was true, but it was very, very strong in the amyloid positive. So it was more tau and the differences were greater. So there was actually an interaction in this graph. So really, she proved her point, but she went on to show that the stronger the functional connectivity, the more tau there was in the brain. So this is really, I think, a nice demonstration that the pattern of connectivity is what's driving the tau deposition in these particular brain regions. But you know, it's unsatisfying because it doesn't tell us why amyloid is driving it or how amyloid is driving it. It tells us that's why it's where it is, but it doesn't tell us how amyloid is driving it. And I'm not going to be able to tell you that. So, you know, forget about that. So now I want to talk a little bit about neural activity and tau because and amyloid, because, you know, I showed you that earlier graph. And this is a functional MRI experiment that we did to try to understand the relationship between neural activity and tau and amyloid. And it's a pretty simple task. What happened in this experiment while people were in the magnet is they saw a stimulus, and then they saw another stimulus, and then the next stimulus could either be a repeat of the previous stimulus like this, so this is the first and that's the repeat, or it could be similar to a previous stimulus, in which case it was a lure. So the answer here is that this is a repeat, it's not a new, and the answer here is that it's new. And so we did this for a series of objects and a series of scenes. And I'm actually not going to go into the real minutia of this task because it gets very complicated very quickly. But I want to first tell you about the first thing that we looked at. Well, actually, it wasn't the first. It's one of the more recent things we looked at, where we asked the question, how is this neural activity related to longitudinal deposition of tau in the temporal lobe? So what we did is we examined brain activation associated with tau deposition in 3 brain regions. One was the entorhinal cortex, that's here in green. The other was parahippocampal cortex in yellow. And the third was the inferior temporal cortex in blue. And we picked these because we thought entorhinal cortex was where tau starts. Parahippocampal cortex was kind of intermediate. And inferior temporal cortex is late. Inferior temporal cortex is really part of this anterior temporal network. And so this is probably, it's an early cortical region, but it's late compared to these medial temporal lobe regions. So we asked, in this paper, Jenna asked, what are the factors associated with tau deposition in these 3 brain regions? So the first thing that she found is that tau in the entorhinal cortex is driven primarily by age. It's not really, age isn't that important in these other brain regions, but in entorhinal cortex it is. And I mean, again, frankly, this is not a new finding. This is, I think, very well replicated. We've seen it in the Berkeley data before. We see it in the ADNI data. Many other people have reported this. So entorhinal cortical tau is very strongly age-related. But what's interesting is that when you look at amyloid, amyloid drives tau in all 3 of these brain regions, entorhinal cortex, parahippocampal cortex, and inferior temporal cortex. But in a multivariate model, it's really most important in the inferior temporal cortex. So it seems that age drives the early phase of tau accumulation in entorhinal cortex, but amyloid drives the later phase of tau accumulation in inferior temporal cortex. So then she looked at neural activity, and here we see activity during that functional MRI task, and we're looking at change. Now we're looking at change in tau over time. I didn't really point that out here, but this is also change in tau. So this is age versus longitudinal tau deposition, and this is amyloid versus longitudinal tau deposition. And that's what we're looking at here. Again, we're looking at baseline activation in these brain regions compared to longitudinal change of tau in each of these brain regions. And the tau accumulation in the medial temporal lobe, again, is also driven by activation. So in both entorhinal cortex and parahippocampal cortex, more activation during that task drives tau accumulation. But interestingly, that wasn't the case for amyloid. In this particular data set, activation in the inferior temporal lobe was more related to amyloid accumulation, not tau accumulation. Okay, so again, this is some evidence that similar to amyloid, neural activity seems to be driving tau as well. So now I'm just going to end with a little bit of data on the consequences of tau deposition, both to structure and function, and to some extent cognition. So let's go back to this task, and I want to look at two things. One is something that we call domain specificity. So objects should be perceived using that AT network. So in other words, there should be more activation for objects in this AT network, in the object network. And there should be more activation for scenes in the PM network. So how does tau affect that? And the other thing we looked at was repetition suppression. And that is a very widely replicated neural phenomenon where there's greater activity for the novel stimulus than for the repeated stimulus. So the first time you see this chair, there's more activity in your brain than the second time you see the chair. And this, we don't really know what repetition suppression is. It may be related to recollection, doesn't seem to be actually, maybe more related to familiarity. It's some kind of signal detection. That's sort of the most specific thing I think we can say. So here's the domain specificity. And it's really pretty simple. So let me take you through it. So we're looking in the PM network, the scene network. And this is the activity in the scene network when young people are seeing scenes and when young people are seeing objects. So this is domain specificity. The scene network activates more for scenes than for objects. These are the tau negative older adults. They still show domain specificity. The scene network is activating more for scenes than for objects. These are the tau positive individuals. This domain specificity is reduced. This network, this PM network, is becoming de-differentiated. It's becoming less specific for scenes, and it's starting to activate more for objects. And this is the statistical comparison that tells you that. So what's happening in tau positive adults is they are losing domain specificity. The posterior medial network is no longer as specific for scenes as it is in young people and amyloid negative and tau negative old people. So we simply created a measure of this domain specificity, or Anna Maas did in this paper in Brain. And what she showed is that as domain specificity decreased, performance on the task went down. So this is the correlation between task performance, how they did on recognizing the new versus the repeated stimuli versus the lures. As domain specificity goes down, their performance gets worse. And in fact, it was even related to their performance on the delayed figure recall in the Wechsler memory scale, a long delay figure recall. So as their object, as this network became less specific for scenes, their performance on visual recall went down. So this suggested to us that there's a real world correlate of this kind of neural de-differentiation. So here's the repetition suppression story. I'm going to skip this because I'm going to skip this because it's 9:45. I want to leave time for questions, and I'm going to end with another study that is a little complicated. So I want to spend some time on it. So I just went through this whole story of functional connectivity. You guys all, I think, have an understanding of what that is, but you might not be familiar with this idea of regional homogeneity, or ReHo. And this is simply local connectivity. So what we're doing in ReHo is we're looking at the correlation between adjacent voxels and simply correlate a correlation coefficient between these adjacent voxels and actually do it on a level of basically 3 by 3 by 3 voxels. And you can actually look across the brain at volumes like this and map out the extent to which the homogeneity in the resting state functional MRI signal ranges from highly homogeneous to less homogeneous. And this is a map of this regional homogeneity in a group of young subjects and a group of older subjects. And there are two areas that kind of pop out at you, maybe not immediately, but I think you'll see it when I show it to you, that in the medial temporal lobe, specifically hippocampus, entorhinal cortex, there's much more regional homogeneity in the older subjects than in the younger subjects. And you kind of see the reverse in the medial parietal lobe. There's more regional homogeneity in the young subjects compared to the older subjects. So, and I'm just mapping it out here, that these red regions are the same regions where a regional homogeneity is going up. So you can see these also are kind of, they include the hippocampus, but they also include to some extent these other brain regions that are part of the anterior temporal system. So Tessa Harrison, who's now who was a postdoc and is now working as a scientist in my lab, tried to sort of dig into this and understand what this was related to. So the first thing she did is she had this idea that what is happening is that the hippocampus is becoming disconnected from the rest of the brain. And we know from sort of anatomical studies that that's true, right? One of the first observations that I think actually is Brad Hyman, who really made this observation, is that tau pathology in the entorhinal cortex disconnects the hippocampus. And so what she did is she looked at the relationship between the hippocampus and the anterior temporal brain regions and the posterior medial brain regions and calculated functional connectivity between those brain regions and looked at their effect on regional homogeneity. So this is the connectivity between the hippocampus and the AT network. And this is hippocampal regional homogeneity. So what's happening is as functional connectivity goes down, in other words, as the hippocampus becomes disconnected from the AT network, regional homogeneity goes up. And she doesn't see that with the connectivity to the posterior medial network. So the hippocampus is specifically being disconnected from the AT network, not the PM network, and that's driving up this homogeneity. Then she looked at the effect of tau on this, and she found that there is an effect of tau, that again, as tau goes up, functional connectivity goes down. So this hippocampal functional connectivity to the AT network goes down with increasing tau in the inferior temporal region in the brain, but also in the whole anterior temporal region in the brain. So in other words, as tau goes up, functional connectivity to the hippocampus is going down. So this hippocampus is getting disconnected from the anterior temporal brain system network, and that disconnection is driving ReHo, but it's also related to the amount of tau in the brain, and that's not happening with hippocampal connectivity to the posterior medial network. This tau in the anterior temporal network has no effect on this. And then finally, this hippocampal ReHo is related to memory. So as the brain gets more homogeneous, as the hippocampus gets more homogeneous, episodic memory gets worse. So we didn't see this for working memory or executive function. So this story suggests that one of the functional consequences of tau in the anterior temporal network is disconnection of the hippocampus and also driving memory loss. Just a last couple of points. One is the effect on cortical thickness. Actually, I think it was you guys in San Diego. I think it was a paper by Raul Desikin and I can't remember who else was on the paper. That was the first to show that CSF measures of amyloid and tau were the most predictive of cortical atrophy. And what we found in our Berkeley cohort is actually that the brain regions, that the effect on cortical thickness is driven not simply by tau, but by tau and amyloid. So individuals who are PIB positive and tau positive, so this is the ATN system. These are the A plus T plus cases. These individuals who are positive for amyloid and tau show the greatest cortical thinning. So again, just to show you some structural consequences of this. And then finally, I think this is my last slide, the consequences on cognition. And, you know, there haven't been that many longitudinal studies of the relationship between tau and cognition. And in this paper that just came out, Shi Chen, who's a postdoc in the lab, looked at tau in the AT network, the PM network, and the entorhinal cortex, and looked at subsequent change in cognition. And so this is the effect of tau in the AT network, in the amyloid negatives and the amyloid positives. So there is an effect of tau that's a level effect, but there's an interaction effect with time such that higher tau is associated in the AT network, is associated with more cognitive decline in this memory composite over time. We also see this in the PM network, and we actually also see it to some extent in the entorhinal cortex. So all three of these brain regions show an interaction between amyloid and tau, again, just like the cortical thickness, that tau is bad, but it's much worse when amyloid is there. And so what you're seeing is a decline over time in this memory network, in this memory composite with increasing tau in AT, PM, and the entorhinal cortex. But when you put all these in a multivariate model, it's AT that's the most predictive. So again, even the entorhinal cortex isn't really that predictive. There's again a level effect, but not an interaction. And what the interaction is telling you is that the presence of tau in the AT network is having a effect on the rate of change of memory loss over time. Okay, so very simply, beta amyloid deposition occurs simultaneously throughout the brain. I think that evidence that it's related to some kind of local tissue characteristic, probably related to metabolism and neural activity, is really more, to me, compelling than any story about neuron to neuron or network related transmission. I think there's something that makes a region vulnerable, and amyloid deposits really quickly throughout the whole brain. It didn't go into the speed of this, but it really is fairly fast, I think. Tau deposits in the entorhinal cortex. There's been a lot written about tau spread from neuron to neuron, but I think if you look at the neural pathways, it explains why tau is where it is in most people, that it's in the inferolateral or anterolateral part of the temporal lobe that parallels the AT network. And I think it's the neural connectivity patterns that explain why tau ends up where it does first. And that this tau, particularly in the AT network, has a big effect on brain structure and function and on cognition. And this whole story is really exacerbated by beta amyloid. So I'm glad to answer questions. This was a shot of my lab. We got a shot where everyone was smiling. And I'm sure it looks like all of you guys when you have your meetings nowadays. These are some of the folks who I mentioned in the talk. I really couldn't mention everyone, but obviously the work is done by a lot of smart young people who I've managed to hoodwink into working for me. So I'll be glad to answer questions.
Bill, thanks so much. That was really awesome. And, you know,